which targets the or which recognizes the galactose ligand. So this is what I exploit for my drug delivery system. That is why I stressed here to bring your knowledge uh, or to draw your attention to this particular part. So because of this principle, I have come up with a hypothesis that a natural polysaccharide called as guar gum, it consists of a linear long chain molecule uh, that is that consists of mannose residues and D-galactose. Uh, so this has galactose residues. The galactose residues in guar gum targets the asialoglycoprotein receptor of liver. So this is my hypothesis. So that is the reason why I chose this nanocarrier. So first as a part of my work, I have done few functional group modifications on guar gum for targeting, which to enhance its targeting property. So first, this uh, was a function that function group was modified to carboxymethyl guar gum. A COOH group was attached here, to which the amino conversion was done using ethylene diamine using carbonamide chemistry. So you can see that guar gum was the functional group modification, a COH group was given here, and also an amino conversion was done on COOH. So these functional group modification enables it to be conjugated to any other ligand or other targeting system. The functional group modifications were confirmed using FTIR spectroscopy. So first, I tried entrapping sorafenib drug, which is currently used for treatment, in guar gum nanoparticles. But so first, I prepared a standard curve of the drug, and the drug entrapment efficiency of this developed nanoparticle system was calculated. And for guar gum alone, the entrapment efficiency was calculated to be only 10%, which is very less compared to a good targeting system. So it might be because guar gum is a carbohydrate, and because it is a hydrophilic molecule, the efficiency of it to entrap a hydrophobic drug, sorafenib, might be less. So that might be the reason why the entrapment efficiency was only 10%. So guar gum was again modified using a hydrophobic moiety to increase its hydrophobicity so that it can entrap, or the drug holding efficiency can be enhanced. It was conjugated to another hydrophobic moiety, which is called as stearlamine. It is an 18 carbon hydrophobic molecule. So because of the functional groups, because of this amine group on stearlamine, it could be easily conjugated to the COOH modified guar gum. So this was the next step which I did, functional group modification. And again, the functional group modification was confirmed using FTIR. Again, I checked for the entrapment efficiency, and it was found only to be 54%. So again, to enhance I also looked into the release kinetics of the stearlamine conjugated guar gum nanoparticles, and there was a sustained release. But again, I opted for a good drug delivery system which could hold the drug, the entrapment efficiency. That is why I selected PLGA nanoparticles, polylactate co-glycolic acids. Again, it is a hydrophobic moiety, so I could attain an entrapment efficiency of 84%. This is the TEM image of PLGA nanoparticles, and particle size analysis was done using DLS. Again, zeta potential, that is the surface charge of the PLGA nanoparticles, were done. And also to retain the targeting property of the drug delivery system, I coated guar gum on PLGA nanoparticles. So PLGA was opted so that the entrapment efficiency could be retained, and guar gum was coated on PLGA nanoparticles so that the targeting property could be achieved. So this was confirmed using, again, taking the zeta potential of nanoparticles after coating using guar gum. So the shift here in zeta potential confirmed the conjugation of guar gum to PLGA. I haven't shown the FTIR here. I also confirmed it using FTIR studies. And the ongoing studies of this drug delivery system, I am yet to do the in vitro studies. Parallelly, I also tried creating tumor in animals, not skid mice. And just for a preliminary study, I tried the efficiency of this drug delivery system in animals, and compared to the control, um, the rate of increase of tumor was substantially reduced in the drug and trap Guargam coated PLJ system. So I am yet to do all the in vitro studies. Uh, so I hope, in a nutshell, this PLGA based nanoparticle system was developed and characterized, and the targeting moiety used was guargam that binds to the acyloglycoprotein receptor of liver. And I hope it will be a suitable liver targeting system to reduce the drug dosage of patients so that it targets directly to the liver. These are the references. I cannot end this presentation without thanking all the people listed here, to the director, M. Radhakrishna Pillai. He was a great pillar of support, and also a big thanks to my mentor who supported me, to all the instrumentation facility of RGCB, CSIR for giving me fellowship, all the friends, all my friends and colleagues and family. So I would like to end my presentation saying this: stay hungry, stay foolish. Thank you.
Thank you, Tina. It was a good presentation. Now the floor is open for QA session. Over to you, Dr. Sharil and Dr. Vinod Kumar.
Thank you, Tina. So it was again a good piece of work.
Thank you,
And
sir.
as you said, you are in a prelim work and you need to go for in vitro studies and in vivo
Yes,
studies,
yes.
right? Yeah. So one of
Yes.
the thing is, I just want to know what is the size of the nanoparticle. Just it was not mentioned as the peak picture was there, but I couldn't make out the size of nanoparticle. Could you just maybe the from the slides I cannot read.
Sorry, I forgot to mention it. It was nearly 234 nanometers, sir. I could
Okay.
not mention. It gives the average. It gives the average
Yes.
particle size of all the nanoparticles in the colloidal solution.
Yes, it's fine. But if it is beyond 200, if it's beyond 100, can we say it as a nanoparticle?
Yes,
I
yes, of
again
course.
suspect so I have that query because sometimes you may go for a publication.
Ah,
There may be queries on this, maybe this particle size. So
ah,
just address
yes, sir.
this when you're going for a advanced paper. So you're
Yes, sir.
according to the
For
journal you're communicating, they may
ah,
have uh, issues on this the particle size.
um,
So just check
yes, sir.
out with.
So the problem, like the size which is above 100, that does not make it a, which is not a efficient nanoparticle, because we have taken, in my other studies,
Yes.
I have conjugated a peptide,
Okay.
and we have done the cell uptake studies of nanoparticles to cells. So
Okay.
the nanoparticles which were of this range, they could be
Okay.
easily taken up by cells. So I don't
Okay.
think a nanoparticle that comes in this range will be a problem to be taken up by cells, because it is taken up using receptor mediated endocytosis. So
Whether
this
my
uh,
question is just like whether it can be said
considered as a
classified as a
nanoparticle.
nanoparticle. Yeah.
Uh, yes, sir.
So
I
just
think
you need to just check out the literature and in even the many of the literature you can say the size range of nanoparticle is just
okay.
between this. Okay, just
Yes,
clarify
I'll look
this.
into it, sir.
Maybe you need to look into it because you have a good piece of work, but just because of
Yes,
this you should not get your papers rejected, right? Yeah.
yes. Thank you,
So
sir. Sure.
do the best, Tina. Yeah, all
Sure,
the
sir.
best.
Thank
Yeah.
you.
Yeah,
Cheryl, madam. Yeah.
Tina. Yeah,
Yes, ma'am.
Tina, good piece of work. I would
Thank you.
like to, yeah, just it. I guess regarding to the size of your particle, probably even if you get now, of course, like we consider nanoparticle, the smaller the particle, it's better. But
Yes.
if the role of the particle or the purpose of for you synthesizing the nanoparticle has been met, I guess might be you can justify the size also. Okay, because
Hmm.
like it
Yes,
is
yes, exactly. Yeah, that
that
is
is
why.
the thing that is the
That,
thing which I would like to comment on that particular thing.
yes,
Okay, but I would
because
could you just explain?
for another
Yeah, could
piece
you?
of my work, we could see that the nanoparticles were taken up inside the cell. In this study, I couldn't do a cell uptake, but in another piece of my work using nanoparticles, which is of this range, of the size of the nanoparticle was of this range, and we entrapped a dye, and we could see that the nanoparticles were taken up inside using receptor-mediated endocytosis. So,
Okay.
like how you mentioned, as long as it meets the purpose.
Yeah,
Yeah.
that's the answer that I would like. But Tina, could you just explain to me what was your methodology for your nanoparticle synthesis? Is that been included, or like can you just?
Yeah, definitely I can explain. Nanoparticles were synthesized using solvent evaporation method.
Yeah.
So here, PLGA was dissolved, both PLGA and the drug. We select a solvent in which both the nanocarrier and the drug dissolve. So here I selected DMSO because sorafenib is soluble in DMSO and PLGA also. So both the drug and PLGA will be separately first dissolved in DMSO, and drug suspended in DMSO will be added to um, the suspension of PLGA, and it will be kept for stirring for 24 hours, after which we take out the supernatant using ultracentrifugation by repeatedly washing using distilled water. And then we sonicate and then lyophilize. So we get
I need to ask you something over here. Okay, so you're taking your solvent extract of the particular guargum, right? That's it.
Come again, ma'am. Sorry, I missed out.
You had mentioned about guargum, right? The
Yes.
solvent